we could get prominent scientists today talking to us, sharing their research. And I got the privilege to introduce the first person today. So it's Jan Huisken. Jan is very well known in the light sheet community or in a bioimaging community for all his contributions to the light sheet microscopy. And he is a prime example of the fact how life goes in a circle. So Jan studied physics in Göttingen and then in Heidelberg. And then he joined the EMBL during his PhD, that was in Ernst Sterz lab. And there, basically, he made this fundamental contribution to light sheet microscopy. He likes to call it selective plane illumination microscopy. That's the term which Jan coined. And then he joined Didier Stanier lab at the University of California, San Francisco as a postdoc. And then I was privileged to work with Jan for 6 years at the Max-Planck Institute for Molecular Cell Biology and Genetics in Dresden, where Jan was the research group leader. Then Jan moved to the US, to the University of Wisconsin, where he was basically head of Department of Medical Engineering in the Mortgage Research Institute. And recent development of his CV is that he got awarded by prestigious Humboldt professorship, and that basically brought him back to Göttingen. And Jan is going to share with us his insight on how to put advanced light sheet microscopy to the hands of biologists.
Thank you, Jan. Thanks for the introduction. Thanks for the invitation. It's great being back in Göttingen. So I see this, of course, a little bit also as an advertisement for us, for the lab. Obviously, we are building up the lab here still. I'm still waiting for the lab to be renovated, but we're also recruiting on all levels. So anybody interested, please contact me. So what I'll talk to you today about is going to be a little bit less about the science we do in the lab, but more about sort of the translational aspect of how to put the microscopy that we develop into the right hands, into the hands of the biologists. So I'll first talk a little bit about how we've used light sheet microscopy in the lab, give you some examples, and then share some thoughts with you about how we think we could do microscope development more sustainably in an optics lab, and then in the end talk about flamingo and shareable microscopy. So let's start by introducing the lab very quickly. So while we may not cover necessarily all aspects of modern imaging experiments, we certainly try to. And that includes that we not only develop the microscopes, but we also think quite a bit about the biology, the sample preparation, all the way to the image analysis. And I think that's what probably ideally a modern imaging lab should look like, that the imaging itself is actually embedded in all the other disciplines that are necessary to make all this endeavor a success. And that's certainly something we are also striving to do here in Göttingen. So the technique of choice that we use is light sheet microscopy. I don't think that needs to be introduced very much anymore. The basic concept is that you have two distinct optical paths, one for illumination, one for detection. You generate optical sectioning directly by shining a thin sheet of laser light into the specimen, and then perpendicular to that sheet, you're collecting the fluorescence. By moving the specimen through the light sheet, you can collect three-dimensional data, and on top of that, you can even rotate the specimen in some setups that allows you to cover more areas of the specimen and do what we call multi-view fusion to do more in total imaging. This is what it typically looks like. So you can see there's, in this case, 2 illumination lenses and 1 detection lens. And here where you see the light sheet, that's where you would place the specimen. So not only by us, but now by many, many labs, light sheet microscopy has been used primarily in developmental biology to study organ development, embryogenesis, all sorts of biological phenomena that I would describe as multi-scale, in the sense that you want to see small details happening in the context of larger tissues or entire organisms. So I'll give you just a few examples here. This is a collaboration with John Bouguer's lab back at the MPI in Dresden, where we looked at spindle scaling in early zebrafish. So we see the very first few cell divisions in an embryo. And then if we move up in scales, this is what the entire embryo looks like a few hours later. And in this case, we are imaging all the nuclei with a histone label. And the color code here is just for depth to illustrate the three-dimensional nature of the data. What's important here to understand is that the data set is about 1 millimeter by 1 millimeter by 1 millimeter. And we are imaging this embryo every like 30 seconds or so. And this is actually a fusion of multiple views. So this already gives you a sense that these are enormous amounts of data. But at the same time, you have to be aware that we are looking only at a single embryo here. And so we are still far away from understanding development, because in order to do that, we really need to look at many, many specimens. And so part of the challenge for the future, obviously, is then to develop some more high throughput techniques where not just the imaging is high throughput, but also the data analysis, which I think is the key challenge for the future, being able to extract meaningful information out of this. So if we move up in scales to something like a zebrafish larvae that's a few weeks old, we realize that this is something that cannot be done alive anymore. So we are spanning the range from in vivo time-lapse imaging all the way to fixed and clear tissue imaging. And both can be done with light sheet microscopy, ideally. So the technique is still perfectly suited to also image fixed tissues, clear tissues, because again, speed matters. You want to be as fast as possible to be able to acquire these large volumes with still high resolution. So of course, the challenge here for the future is to push the limit further and further, being able to image longer and longer alive. And then at the last time point, basically fixing and clearing the specimen. And then ideally, which is one of our goals, is to merge the data. So you have the maybe lower resolution, low spatial resolution, high temporal resolution in vivo data set, and you have a high resolution, spatial resolution data set at the end. And you basically want to animate that tissue and use the time-lapse information to go back in time. So I think that's part of one of the challenges, to be able to do this multimodal imaging using different inputs, ideally extracting as much as possible from one specimen. So just trying here to summarize some of the take-home messages. So first, light sheet microscopy offers both high speed and low phototoxicity. And I think these are the two key elements that allow us to do some of the experiments that I'm going to show you today. The other aspect is that a lot of what's really critical for the success of an experiment is that you can custom build a microscope and tailor the microscopy around the specimen. And I'll give you a few examples for this. And I think this is sort of a general theme throughout the talk or throughout the entire lab, that we see a lot of value in being able to customize every single aspect in a modern imaging experiment, from building the instrument to writing code to analyze the data. So one example is going to be cardiac morphogenesis and function, something we've done quite a bit in the lab. So here the goal is essentially to image this organ, the zebrafish heart, in 3 dimensions, or actually in 4 or 5 if you want. So doing multicolor imaging in a 3-dimensional object that's also moving very rapidly. And over the years, we've perfected these tools. This is actually data we took back in Dresden with Michael Weber. Here you can see the result of such a 5D reconstruction. So we're looking at a projection of a 3-dimensional data set in 2 colors over time. And the key to success here is that the heartbeat is very regular. We also have techniques where it doesn't necessarily need to be regular, but in this case, we could exploit the fact that it's very regular. And so the technique here is the following, that we take movies, as shown here on the right, at a certain depth throughout the heart. And we can take these movies at about 400 frames per second in two colors simultaneously. And then if you've taken movies at different depths throughout the heart, you can synchronize these and reconstruct the heart in 3D as it's shown on the left. And so over the years, we've taken data sets like this to be able to understand and describe in detail, for example, the mechanics of the heart. So in a recent project, we're trying to understand how the myocardium contracts and how this force is exerted on the endocardium, the inner layer of the heart, and how this is folded and which role the cardiac jelly, the incompressible extracellular matrix, plays that sits in between these two layers. And so that allows us then to develop three-dimensional models of the heart. And we even built like a macroscopic model of the heart to replicate some of these mechanical features that we observe in the zebrafish. Another example for this, where the customization is key to success, is the attempt to describe zebrafish gastrulation. So in this case, we are essentially trying to acquire data that I just showed you earlier of the entire embryo, trying to track essentially every single cell. But as I said, if you do that, you are accumulating so much data that it becomes almost impossible to do this over many, many embryos. And so in this case, what we did is we designed this custom microscope that has 4 objective lenses, 2 illumination lenses, 2 detection lenses. And as we move the fish through the light sheet, we're actually we are only saving a certain amount of data, only the relevant amount of data, and not the inside of the embryo, which is empty, and not the outside the embryo. So what we generate in this case is a maximum intensity projection, which is shown here, a radial maximum intensity projection, which is represented in a 2-dimensional image at the end of each stack. So we are throwing away pixels that we don't care about and generate this Mercator projection on the fly as we're taking the data. So what this means is that at the end of the time lapse, you don't have 4D data, but instead you only have a little QuickTime movie that shows you this projection of the entire embryo like a world map, and you can very nicely visualize the migration of these endodermal cells. And this also means that the analysis and the visualization of the data is so much easier that it's actually a step towards doing this in a high throughput fashion and also being able to characterize the phenotypes more precisely. So moving up to multicolor data. So if we're not just looking at the endoderm, but the other germ layers as well, we're looking at 3-color data. And in this case, we don't do the maximum intensity projections anymore, but instead we've developed tools that allow us to visualize the tracks of these cells in a more general form. So in collaboration with Nico Scherf, a former postdoc in the lab, we've developed this tool that allows us to visualize the tracks or tracklets, the smaller tracks of these cells in a way, and we can also bundle these data more efficiently. So instead of looking at this noisy data set, we can identify where the cells are actually mainly moving and condense these tracks onto these highways. So we can see which direction the cells are primarily taking, and we can also unfold the data again in this Mercator projection to reduce the dimensionality of the data. So this basically shows you that in order to do an experiment like this, you ideally have a home-built microscope, you have written your own code to run these kind of experiments like the radial maximum intensity projection, And you also need custom software for the data acquisition and the analysis. Another aspect that's really important for us is that this whole process ideally should be reproducible. And with that, I mean reproducible not only by other people, but ideally also by your own people in your own lab. And that often is tricky because sometimes, or what's often the case, is that you have a postdoc join the lab, they build up a fancy microscope, they publish, they leave, and then nobody knows how to use the instrument anymore. And obviously that's not very sustainable. So that brings me to the second part of my talk. And this thought process was mainly triggered when we moved from Dresden to Madison. We were looking at all the instruments that we have built and all the projects we had done. And we realized that the 8 microscopes, the 8 light sheet microscopes we had home built in Dresden were almost impossible to move to a new place, because as soon as you disassemble them, nobody knows how to assemble them anymore because the people had left the lab already and nobody would know how to use the software and so on and so forth. We also realized that we've mainly used the same components every time. So as you can see, blue is always the illumination lens, green is the detection lens, there's always a camera, there's maybe some additional features that make this instrument specific, but in the end, ideally, you would always want to use the same component. You would want to have some sort of a Lego set for a light sheet. So one example here is a rather standard T-SPIM, so a 3-lens light sheet microscope with an LED for transmission. And in the lab, we use this system to do OPT, optical projection tomography, together with light sheet microscopy. And it allowed us to collect, in addition to the fluorescence, also the rest of the fish, and it turned out to be incredibly helpful for analyzing and visualizing this data. This, in fact, isn't really a major change in the optical setup. It's rather more in the software. So you could use a regular light sheet microscope to do something like this. Another example is the magnetic rotation of samples that we developed. In this case, again, we use a regular T-SPIM, but in addition here, we are using an array of electromagnets. And the reason why we use these electromagnets is that we developed a technique that allows us to inject magnetic beads into embryos, or sometimes even just into the yolk cell of a zebrafish embryo. Because once you've done this, you can use, in this case, like a permanent magnet to rotate these specimens. And so if you have electromagnets around your specimen in a microscope, you can turn these on and off, and that way you can rotate the specimen in the microscope, which again helps with the multi-view imaging. But also if you don't necessarily want to do that, it already helps orienting the specimen in the microscope to get the perfect angle, the perfect view to get the best possible data. So looking at these instruments, we realized that they are not necessarily terribly complex, but they are not really movable. And so if we had only built them on like smaller breadboards, we could probably lift them up and pack them and send them somewhere. But it would still be very complicated with all the cables and all the electronics and so on. So we had to come up with something better than that. So over the last few years or so, in Madison, I was fortunate to have 3 dedicated engineers. So Rory Power developed all the optics, Todd Bucken, a prototyping engineer, developed all the mechanics, and Joe Lee wrote all the software for what we now call the Flamingo. And so the idea here was to come up with a modular design that allows us to basically replicate all of these different setups that I explained to you in a more streamlined fashion. And so what was born was this Flamingo, which happened to be also very compact. So the instrument itself has the same performance as what we used to have when we built instruments on an optical table, but at the same time, it happened to be so compact that you can easily put it next to like a stereo microscope, as in this case. And most importantly, it was modular in a way that we can build multiple different light sheet configurations using the same components, which also meant that if you start one day using a certain configuration, you only needed very few parts to convert it into a different configuration. And that, of course, is incredibly powerful because you don't necessarily know in advance which configuration may be best for a certain application. So here's just two systems side by side as they are being built in the lab. And the other thing you recognize here in the back are these electronic boxes. And so we found a way to put everything we need in addition to the optomechanics and the optics into a single aluminum frame. And so essentially the entire microscope consists just of the optics and the electronics part. And this also includes, you may recognize the Toptica laser engine with 4 laser lines to feed the laser light to the microscope. So here's just a quick walk into the lab back in Madison. So you can see there's actually a total of 4 flamingos here in the lab. One is here on the left in a tent. So that helps to block the light and also for temperature control. And then there's 3 flamingos here on the right on one optical table. And the neat thing here is, again, that you can use different configurations, and you can also build all of these configurations for relatively small amounts of money. And you can reserve the expensive components for whatever system you actually want to use. So basically, you can come in the morning and say, all right, today I want to use this configuration with these lenses. And all you have to do is attach a camera, plug in the fiber, and attach the stages, which are the 3 most expensive components. And then the rest of the instrument is already there, ready to be used. So this is a T-SPIM configuration, 2 illumination lenses and 1 detection lens in the classical light sheet configuration. And then there's other configurations here on the right. This is what we call a V-SPIM in the center, where we have 2 lenses dipping into a Petri dish from the top. This is a 25x 1.1 NA lens for high resolution imaging. And then here on the right is another T-SPIM fully equipped with perfusion, heating, and the stages on top. Okay, so in the last part, I'll tell you a little bit about how this flamingo concept could actually be of benefit, not just for us, but for more people. So during the pandemic, we realized how important it is that you can take these instruments anywhere you want. And this was actually my home office in Madison. So I was able to take a flamingo with me. It was sitting on my desk at home. And we realized that other people in the lab can also run experiments on this instrument remotely. So this was actually part of the software design, that the instrument itself does not come with a computer. You have to bring your own computer, and then you can control this instrument. And this can also be done remotely. So this was then the next step in the evolution, that we realized that the microscopes are portable and we can bring them to the biologists. And this was very much in line with thoughts that we had earlier already, which are described in this comment here in Nature Methods in 2019, putting advanced microscopy in the hands of biologists. So here we describe the idea, how important it is that the microscope is where the specimen is. And that way you don't have to travel with your precious specimen to the developers, right? Of course, maybe you have to go across the street to a facility, but ideally you don't want to travel all the way to the physicist who may have developed a fancy new microscope. The benefit for us in this process would be that we can iteratively pass the instrument back and forth between the engineers and the biologists, and that way optimize the instrument for the needs of the biologist, And at the same time, the engineers would learn what exactly the biologists need. So we're not just developing some super cool microscope in our basement that maybe nobody needs, but in fact, we are working hand in hand to push your science and being able to, push the envelope of what light sheet microscopy can do. As you've seen, the electronics box is pretty compact. So this houses all the controllers for the stages, a mini computer for the control. See a network switch here. There's also a UPS power conditioner in here, power supplies, and the laser engine. And then in the other Pelican case, we have the microscope. This fits into a regular car and can be shipped very easily. And again, the goal here is to bring the microscope as close as possible to the specimen. And ideally, you would want to set it up right next to the fish room or in the fish room so that you don't have to go very far with your specimens. So just a quick cartoon to sort of illustrate how we envision this to work on a larger scale potentially. So let's say you have an idea how to use light sheet microscopy. You may not, maybe you haven't ever used light sheet microscopy and you got curious. The idea would be that we discuss the project and we build a system specifically for your needs. It could be a standard system or could be customized. We send the instrument to the biologist and then they get to test it. Once they are done, they pass it on. So it's also this idea that if you stop using an instrument, it doesn't sit in a facility or sits in your lab and collects dust, but instead it gets moved on to the next person in line. And of course, it's also being shipped back to us for maintenance or for any modifications. So we've exercised this quite a bit already. We have a system in San Diego. There's one system at Harvard. There's one at MBL in Woods Hole right now. And we've already imaged plenty of different specimens, not only zebrafish, but also this is a Xenopus in Bill Beman's lab, really exciting project. So it works. We can take these systems out of the optics lab and place them in other labs and collaborate with these people. The main important thing I think here for us is to bring the microscope to the biologist. It needs to be where the sample is, but at the same time, the analysis and the image acquisition can be done by someone else. And that's very important, I think. So what you see here on the left is essentially the microscope and its mini controller. We connect wirelessly to the microscope with our laptops. So when I walk into the lab, all I have to do is open my laptop, start the Flamingo software. I can select any of our flamingos from a drop-down menu, connect to them, start an experiment and leave. And then the microscope runs autonomously and acquires the data. Many people can connect to the same instrument, so we can remotely collaborate and design the experiment together. The data is either saved locally on a USB drive or your file server, but it could also be streamed to a cloud. And then another person can grab the data and process the data. And that also allows us to then give feedback to the instrument and change the experiment. So it essentially is an idea for like 4 people collaborating, or 3 people, let's say. One person prepares the specimen, puts it into the microscope. The second person, who's the microscopy expert, runs the experiment remotely if you want. And then a third person does the data analysis remotely and gives feedback to the other 2. And this, of course, is also in line with this idea of smart microscopy, where you have the ability to analyze the data on the fly and then give feedback to the instrument to acquire meaningful data, increasing the information content in the data and acquiring less redundant data. So the microscope should be where the sample is, but the data acquisition and analysis could be done remotely by other people if you want. So that, I think, is, the more we think about it, we think about a lot of cool ideas where, you know, especially like in the zebrafish community, you don't necessarily need to have the fish sent to you a specific transgenic line you want to image, but you could also send them the microscope and have them place the sample in the microscope. And you could still run the experiment if you want. Or you could have other people run experiments in your lab. So let's say in the evening when you go to sleep, some other person may wake up and use your instrument remotely. And so that way we're using these resources more efficiently. So again, this is, I think, a great idea towards more open science, more reproducible science, because as soon as somebody has taken data on a flamingo, somebody else could request the same experiment and reproduce the data that way. So with that, I want to acknowledge people in the lab. This was my lab in Madison and a list of recent collaborators. And specifically, I want to thank people in the lab that have focused primarily on the Flamingo project and these companies for their support. And I would invite you to go to the Involve3D.org Flamingo website, or Huiskenlab.com works as well, to find out more about Flamingo. And again, as I said in the beginning, we are starting a new lab here in Göttingen and recruiting on all levels. So if you're interested, please let me know, and I'm happy to take any questions if we still have time. Thank you.
Yeah, thank you so much for this great guided tour on your achievements there. Did flamingos already fly together with you to Göttingen, or they are still in the US?
They are not yet here, still not here. We are still finalizing the purchasing process to buy some flamingos from my old institutions. But the mechanical workshop here at the Institute, which is fantastic, by the way, has already started machining some of the custom parts, and we are placing orders for cameras and lasers, etc., to be able to build these flamingos here very soon.
Thank you so much. I would ask audience, for time reasons, possibly 1-2 questions, because then we would need to move on. Jan, would you still stay with us, that you could be available in a chat, for example? Or
sure, absolutely. I'm happy to do that. Yes.
So feel free to either type the question or just raise the hands and ask Jan.
There is a question in the chat from Gerard Holst. Thanks, Jan, for the very informative and nice presentation. Any plan to combine SIM with Flamingo?
Not yet, but of course the system is flexible, that we can combine it with different techniques, and especially if these are techniques that we are not necessarily experts in, we would of course invite other people to team up with us. We are happy to share, you know, the hard and software with those people and place a Flamingo on their site, and they could develop some add-ons or specific, you know, additional techniques on those systems.
All right, yeah, thanks so much. I guess for, yeah, one more way. Any photo manipulation capabilities? That I would be last question. Now I apologize for the rest. So any, Britta ask, any photo manipulation?
Yeah, of course, this is, again, also very easy to add on. Right now we have a system in Madison that also has a DMD photo manipulator for local photo activation and patterning that we use. So that works really well. But no laser cutter or so yet, but that could be added very easily.
Okay. Thanks so much. I think Meta then do one the offline your question, and I think we need to now give the time to next speaker. Thank you so much.
Sure. Thanks.